This is a production of Cornell University.
Okay, thank you so much for such a nice introduction. I'm really happy to give a talk and talk a little bit more about both my research and the Phenosite, and hopefully I get to convince you all to come and use our fantastic phenotyping facility more, because we do have space and we do have, availability for phenotyping all of your plants. So, as you probably are aware, the world is experiencing crises of all kinds, and agriculture in particular. Let's focus on the crisis that we can actually know something about and do something about. So you can see all kinds of crises, including pollution, deforestation, erosion, things like that. I don't really know too much about soil, which is weird for a root biologist, but yeah. But what I do know quite a lot about is water stress. So you've got, and the blue dots are representing regions that are affected by water stress. So you've got either too much water, too little water, or the wrong kind of water. And that's the case in salt stress. So what we can do about it as plant biologists with our talents, our skills, our knowledge, we can diversify the agriculture. So don't be fooled by my Photoshop skills. Here is a desert rose that is native to an island off the coast of Yemen in Socotra, which is here, very desert-like environment. And we can learn about, from those extreme plants, how are they dealing with those extreme environments, and maybe include that in our species that we like to eat, like tomatoes. Or we can look at the species that are very closely related to the species that we already like to eat, and look at the intraspecific variation, so within the species. And that's really the approach that my lab has been taking, to really look at the wild relatives to crops that we like to eat, and learn from them, because those species haven't been pampered in the greenhouse, haven't been pampered by the breeders, so they really had to fend for themselves. So let's learn from that. And so, as Jan already mentioned, so we're working on quite a variety of species. Arabidopsis is still a workhorse in the lab because it's just an easy plant to work with and validate some of our things. We're also working on the wild tomato, which is a very close relative to cultivated tomatoes, Solanum pimpinellifolium. So that's this guy over here. We also have been working on tepary beans. Tepary bean is a close relative to common bean, so it's a Phaseolus species. It's native to southwest of the US and northern Mexico, so another very dry environment, and it has been used by native communities for centuries right now. We've got natural diversity panel established for that we have sequenced. I'm not really going to talk too much about that. The other species that we're also investigating is cowpea. So cowpea is native to sub-Saharan Africa, where it has been, a great resource for many subsistence farmers that don't have access to irrigation, don't have access to a lot of fertilization, and it's really thriving there. And the really cool thing about comparing those two species together, tepary bean, it's Phaseolus acutifolius. If you know any Latin, like not me, you can say like, okay, so acutifolius is really because the leaf, the needle-like leaf, right? So tepary bean, it has those very, very narrow leaves. Like, no wonder you're drought tolerant, because you have hardly any leaves. Cowpea, on the other hand, has leaves as big as my head. So it's really interesting that both of those species have drought tolerance, but probably the mechanism or their strategy of dealing with drought stress is very, very different. And at the beginning, when I was starting a lab, I'm like, okay, I'm going to screen all of those species for drought tolerance and see which genes are common between them. And now I'm like, I cannot do that because it's a stupid idea, because all of those different populations are very different. So cowpea, we're really screening a population of domesticated species. Tepary bean, we've got a mix of domesticated and wild accessions. Solanum pimpinellifolium, as far as I'm aware, except maybe for Joyce van Eck, nobody has domesticated pimpinellifolium yet. So we're really dealing with a variety of different populations and their nature. So of course, we're not going to find the same genetic signals. It's already hard to reproduce GWAS between two different greenhouses, so let alone between the species. But in the end, I'm like, well, it might be not the best idea to look for commonalities of drought stress resistance, but diversity, and that's a hot word in the recent climate, but diversity of mechanisms and strategies of dealing with drought stress might be actually even better, Because if I have my own ways of dealing with stress before the presentation, and maybe Jim will have a different strategy of dealing with stress. And if we're kind of exchanging those strategies between each other, maybe we can get even more relaxed and even more chill, because I know that Jim is a great presenter. So similarly with drought stress, maybe by combining different strategies exhibited by different plants, we can actually get like super drought tolerant plants, or breed plants for different drought scenarios, because droughts in Ithaca is very different compared to drought in Arizona or drought in Australia, right? So before I go into more detail, I'm really going to focus a little bit on the discovery pipeline that we're using. So we're really looking at starting with the trait identification. So you have a whole phenotype of a plant, and you can study all kinds of things. The longer you look at the plant, the more things that you can measure you can think of. The trick is, for us, is that that specific trait needs to have some kind of heritability, because we are using forward genetics. If there's no heritability to it, maybe genetics is not the best way of studying it. Maybe biochemistry or transcriptomics is the best way to do that. But for us, we're really relying on genetics. So we're looking at heritability. The other thing that a lot of geneticists are not a big fan of is we're looking at how that trait is plastic in response to stress. So a lot of the research that has been already done on genetics of stress resilience has been done on genes that are like traits that are not necessarily changing throughout the environment. And there was a very good reason for that, because you want your corn to be growing very well in the field in Ithaca, as well as it's growing in the field in Geneva, as well as in Pennsylvania and everywhere else in the world. So you want something that has really, really high heritability. But we're missing on the things that the plants are incredibly plastic. We know that if you grow plants in different day lengths, they start to develop different number of leaves. The leaf size might change a little bit. The number of flat roots is changing constantly depending on the water availability. So we're really interested in like, okay, what are the traits that are showing some plasticity? And that's a part of like an experience of working with GWAS. We really want to have as atomic value of a trait as possible. And that's why we're working with the phenes. So phene to phenotype is the same as gene to genotype, right? So phene is like a dissected component of your phenotype. And if you think about like, okay, well, if I study drought stress tolerance and I just study survival is extremely complex trait, because it depends on how deep are your root growths, what is your water use efficiency, how big is your shoot to root ratio, what is your growth rate maintenance, all this stuff. Are you slowing down as soon as you're like thinking like, okay, I'm experiencing drought, I need to stop for a second and think and adjust and then keep on growing? Or am I just like insensitive to drought and keep on growing, and then I grow myself to death, really? So we're trying to dissect the phenotype into as many as many components that are as easy as possible to describe with a number. And hopefully by doing that, we're increasing the heritability. So if you think about like the heritability of survival, it's hard to measure, but heritability of growth in 1 week after germination, well, that's already increasing, right? And the growth rate is already a complicated trait, and you can divide it even further, right? So then, okay, I spend a lot of time on that. Now that we've got the trait identified of interest, we're going to do a population screening, we're doing phenotyping and forward genetics, and then only once we are at the level of gene of interest, we're doing a study, we're validating it using reverse genetics. We're identifying what is the molecular context, because some of the genes might not be conserved in all of the species, but the molecular context in which they are acting might be easily translatable across different varieties of species that we like to eat. And also at this level, we're really going to look at like, what is the relationship between my trait of interest caused by that one gene and the overall plant performance. We're not looking at the correlations or relationship at the level of natural diversity panel, because those plants that are different in number of lateral roots, number of branches, they are also very different in their root physiology and morphology and pollen development and stuff like that. So if I'm harvesting all of the diversity that is really related to the root system architecture, why should I be ignoring all of the diversity that is happening in the reproductive development, right? So I should be cognizant about that, and looking at the relationship between that at the natural diversity panel might not be the best idea. If you've got maybe a biparental population, that's a very different conversation. All right, so as Jan told you, I already started my kind of research in stress-induced changes in plant architecture, really looking at Arabidopsis, because that's a well-domesticated species in the lab. And really we were asking like, okay, so when I grow my plants in the increasing concentration of salt stress, my plants are getting smaller. But are they growing different? Is just smaller, like, and how are we going to tell if it's just slower and smaller, or is it different? So what we have done is we really dissected the root system architecture in 3 components. So you've got a speed at which your main root is growing, a speed at which the lateral roots are popping up, and the speed at which the lateral roots are growing. So now we're using the same functions for all of the 3 components. If all of my functions are decreasing by 50% under stress, it means the root system architecture is not changed, it's just really slower. But if one of my functions is decreased by 20% and the other one is decreased by 80%, it's like, okay, the stress is really actively reprogramming my plant architecture, my root system architecture, because the plant is actively choosing to put more biomass in their lateral roots compared to the main root. So those models were the basis of the whole screen of the natural diversity panel back then that we had in Arabidopsis. Right now, the natural diversity panel of Arabidopsis is extended beyond very Eurocentric populations. So we have collected some accessions from Africa, Saudi Arabia as well, and Greater Asia. We have also developed this screen to look, because root system architecture, we're a very proud bunch of people because we're working on a hidden half, but it's still a half of the plant. So when I was doing my postdoc in KAUST, Mark asked me like, well, what happens to the root to shoot ratio under salt stress? And so we developed this little tool that you can quantify the shoot size and the root size from your agar plate images. And we realized that, okay, the roots to shoot ratio, unlike under drought stress, under salt, it just gets dysregulated. We identify a gene that is causal for that. And that paper just got published. So yay. And Mariam, a postdoc in my lab, really made the final strides in that work. But today I really want to focus on Pimpinellifolium. So Pimpinellifolium is a wild tomato. Here you can see a plant of Pimpinellifolium growing in the high throughput phenotyping facility in Australia. So we are working with approximately 220 accessions of Pimpinellifolium that were collected around the west coast of South America and some of the Galapagos Islands. They're an important source of genetic variation. Their native range is really varying because you've got regions that are called the lomas, which are those fog deserts. So there's hardly any rain, and really only the fog is coming in there. But then some of those accessions were collected around the riverbeds, where it's not exactly like limited in water. So we thought, okay, there is a really great variety of environments that those accessions were collected with. So we have already screened them back in KAUST in the field for control and salt stress, and in the plant accelerator in Australia. And that work has also been published last year. So me being me, I'm like, let's have a look at the root system architecture. How are we doing that? I just did that before I left Saudi Arabia. So we're germinating the plants under control conditions. We transfer them for control and salt plates. And then we scan the plates every single day. And then what we're doing is not necessarily high throughput phenotyping. I like to call it high dedication phenotyping, because there is, until now, there was not really a way of automating it fully. And every single person that has been working with half automated pipelines also can appreciate how much work half manual work is, because you have to still like draw all of those roots and make sure that every single time that they are crossing to manually correct like which root is what. So we have done that together with Hayley Sussman, who was a technician in my lab. We have screened the entire diversity panel. All good. But with Mariam, we really looked at the diversity panel in a slightly different way. And we're like, okay, are there any plants that are showing exactly the opposite response to stress? And we have identified 2 lines. So we've got M248, which is Pimpinellifolium. And we thought, well, how about we also look at some of the cultivated lines? So we looked at the LA1511. And as you can see in here, they're showing, both of them are grown on salt stress, and they're showing exactly the opposite response. So one of them is really investing in producing new lateral roots, but those lateral roots, they are not really like very good at growing. The other one, on the other hand, is really very good in investing in elongating the existing lateral roots, but there are not many new lateral roots showing up. So we thought, okay, this is really cool. Maybe we can do some transcriptomics, but which time points? So for that purpose, we grew our plants on this device that is called Spyro. It's entirely, well, it's 3D printed, and it also has some aluminum profiles. So we can image, we can take the pictures of the plants growing on the agar plate every hour. So Mariam or me don't have to sit in the lab and scan those plates every hour, which is, as you can imagine, quite laborious and boring. Anyway, so we collected all of those images. So now we've got a time series that is incredibly dense for both of our lines under control conditions and salt stress. So salt stress is red. And you can see that one of our lines is indeed, so in here we're looking at the lateral root length, individual length of lateral roots. And you can see that one of them is really suffering in their lateral root elongation, and the other one is like, salt? What salt? What's happening? The same happens if you calculate the root growth rate. So it's basically derived from here. And you can see that the root growth rate, again, for one of them is really suffering. And for the other one, it's like, well, no, I don't really see much of a difference. Only at the later stage, we're starting to see difference. Then we looked at the popping up of new lateral roots. And you have in here, you also have the time after transfer, 2 different lines. And you can see in this line that all of the lateral roots, so the line that was really struggling with the lateral root elongation, you can see that the lateral roots are popping out throughout. If you look at the average, yeah, it is a little bit delayed, but average in that specific scenario is not super informative. But if we look at the other line, you can see that all of the lateral roots that are indicated by the red bars are clearly delayed in here. So this super detailed time point allowed us to select time points before we start see visual differences that we then harvested for the roots, for the RNA-seq experiments. What Mariam also did, because she has a training as a reproductive biologist, so she's not afraid of crossing plants. So she crossed those 2 lines, and we looked at the root system architecture of the F1 hybrid. And I was like, well, you know, combining those 2 traits is just too good to be true. And yet, those 2 strategies seem to be complementary, because we don't see plant with either more lateral roots or longer lateral roots. We see both of them. So yes, we can significantly improve, although both of those 2 lines, whenever we look at them in the field, in the plant accelerator, both of them are growing pretty well. We can still improve on the pretty salt-tolerant accessions by combining their complementary strategies, which I think is really, really cool. So what we did, of course, we did all of the genome-wide association. After Mariam crossed the F1, we also grew them out for F2, and Zhang Junfei helped us with the bulk segregant analysis for all of those bulks. And Liang Yu from Andrew's lab helped us with the RNA-seq experiment and analyzing all of this data. So we have the genes that are associated through GWAS. We've got the regions of the chromosomes that are associated with the bulk segregant. And then we've got individual gene expressions that are differing between our lines. So what is the overlap? So we found a couple. There are some of the lines that, or some of the genes that are the DUF 1677. It's one of the strongest candidates, but the DUF stands for basically domain of unknown function. So thank you for calling it anything, but we cannot really like make too much sense out of that. 2 other things that we found that are described as something is this ethylene responsive transcription factor, TO3, and ascorbate peroxidase, so basically vitamin C peroxidase, so having something to do with reactive oxygen species. A couple of months ago, Ron Mittler was visiting Cornell, and Mariam went to his talk, and then she started, sending him messages like, hey, Ron, can you image the roots of our tomatoes? Because we think there is something going on with different reactive oxygen species reactions. So Ron was like, okay, cool, send me the seeds. So we sent him the seeds, and Santosh did most of the experiments for growing the seeds. And basically, here's an example of how, so they are really using a visualization for the hydrogen peroxide in live images. So what we have seen, and in here we've got accession number 1, accession number 2 in root and in shoot. Wait. Yes. Okay, this is complicated figures. So we've got accession number 1 is having much more reactive oxygen species accumulation quite early on in the root, while the accession number 2 doesn't. Like nothing is happening in the root whatsoever. But this one is having much more reactions in the shoot compared to the root, compared to the other accession. Right? So we have the ones that are, if you don't have a reaction in the root, you will have a reaction in the shoot. And it's not necessarily sequential, because in the shoot, we see the signal building up much quicker than we see in the root. So there are the absolute differences in the reactive oxygen species accumulation between those two lines. We're still cloning the genes, and we're hoping to knock them out in both of the lines to see what is the effect. But this is an intermediate result. And then what we also did is, this is another complicated graph. We also grew the plants with ascorbate and with H2O2. So what we're seeing in here, so let's have a look at the salt stress. So the red dots, and we've got the main root growth rate, average lateral root growth rate. So how fast is the main root growing? How fast are the lateral roots growing? How quickly are new lateral roots popping up, right? So this cultivated one is, we've got, okay, lateral root elongation is suffering more than number of lateral roots. And in here, we've got an opposite pattern, right? So we've got lateral roots keep on elongating, but the lateral root number is suffering, right? Cool. So then when we add, in here, the green one is we add ascorbate. As soon as we add ascorbate, this pattern is not really changing much, because the lateral roots are still growing great. We get a little bit more lateral roots popping up, but in here, we are reversing the pattern. So we are rescuing the lateral roots can still grow faster. And then whenever we add hydrogen peroxide into the plates, we're kind of making everything even worse. And this line, for whatever reason, is like, oh, I'm still fine, because it's probably not related to ascorbate metabolism at all. And we also looked on ion accumulation, and we have noticed that addition of the ascorbate is lowering the potassium to sodium ratio. So potassium to sodium ratio is important, because typically under salt stress, potassium is outcompeting the sodium. No, other way around. Sodium is outcompeting potassium. So the higher that number is, the worse the plant is getting. So getting it lowered by ascorbate is a great result, and it's reflecting kind of the changes what are happening in the root system architecture. All right, so that's where we're at with our molecular biology, following up, finding the genes. We also started to explore how we can describe the root system architectures using different metrics. So when we were spending entire pandemic looking at the roots and tracing them, we started to observe a very distinct pattern. So you've got roots that are really growing like a Christmas tree, or roots that are growing like a telephone pole, a little bit droopier telephone pole, and a broomstick. So all of the lateral roots are coming from the base. And in here you've got, it's really like a maze root. Like all of the roots, they are very nicely spaced, but very definite length. We don't have that diversity of the lateral root length as we see in the Christmas tree, right? So we're like, well, this is cool, and I see it, and I know when I see it. But when I'm using my metrics of lateral root number per plant, average lateral root size, even the branch zone size, I always have this one comparison that is not significant. I know that I should have a number. How do I describe that? And that's a never-ending conundrum of anyone that is doing phenotyping. I see it with my brain. how do I translate it into a number? So I was like, oh, maybe I can combine all of those three numbers, mathematical modeling. And then Arjun Chandrasekhar, who is a good friend and a mathematician, he's like, well, no, how about we just calculate the Pareto front optimality for those root system architectures? And then you've got all nicely separated things. I'm like, great. Can you explain what this Pareto front optimality is? I've got a number, but how does the number work? So Pareto front optimality is really designed for like evaluating efficiency of infrastructures in the cities. So in here, we've got a subway network of my favorite city in the world, Amsterdam. And you can see that you've got all of those neighborhoods that are semi-affordable to work with. Everyone needs to go to the central station. And you've got all of those hubs. Sometimes they're crossing over. Okay, we can also think about like that in the root system architecture. So you've got the roots, you've got all of the absorption happening of nutrients and water. Although if you talk to people that are working on modeling of water and nutrients, we still don't really know how the root is absorbing water, which is mind-boggling for me. Anyways, but let's assume that we are absorbing majority of water and nutrients with the tips. And here we've got big pipes that are doing majority of the transport. Okay, so we can think about it as neighborhoods that need to get to central station. So we need to abstract it. So abstracting the root, we've got a root system architecture, but we can connect all of our lateral roots in a very fast way, in a very cheap way, and in some intermediate solution. So basically, what Pareto front optimality is saying, you don't have one optimal solution for your problem. You've got a front of optimal solutions, and you can see how far from those optimal solutions you are, right? And that gives you a number. So, however, there are a couple of issues with the Pareto front optimality. So right now, we are assuming that conductivity and the cost of a lateral route is exactly the same as for the main route. So in here, this is a very quick experiment that I did for this grant that we got, is looking at the number of xylem vessels in the main route compared to the lateral route. And that's the main pipes that are doing the transport. And you can see the blue things. There's huge difference in the xylem number and the surface of the xylem vessels that are doing the transport. So that's already an assumption that is wrong that we need to correct. And also the number of the cortex layers and all of the cells that needs to be not only built, but also maintained as the root system architecture is growing. So right now what we're doing is so Emily and Trent have been really amazing in collecting all of the root sections for the main route and the lateral routes. Those are the best pictures I could get. It's very hard to kind of get them really nice. And we're quantifying basically all of the vasculature, the surface area of the cortex, the endodermis, epidermis, and the xylem bundle sheet cells. And we can see that between the lateral root and the main root, there are tremendous differences. And also there are differences in the developmental stage as well. So your xylem bundles are looking very different at the root tip compared to the very mature root. And at some point, we also are starting to have the secondary growth, which is specific to dicots. So right now, together with Arjun and Guillaume Lobet from Belgium, we are integrating all of those physiologically quite relevant features into our Pareto front optimality metrics. And then using those adjusted models, we're going to recalculate everything for our entire diversity panel and then use that to run the genome-wide association. And hopefully we're going to learn a little bit more like which genes are involved in optimal root system architecture or optimizing the cost or the transport rate. The other thing that I want to just briefly mention, so it's a happy news. Recently, we have been awarded together with a bunch of really amazing collaborators from BTI, this FFAR grant. So we've got Sylvia Restrepo, Jim Giovannoni, Julian Fay from our technology transfer office, Gagan from Myogenics, and Zhang Junfei is really like the lead brain in here. And basically what we're going to do is we're going to screen the wide tomato diversity panel for drought stress tolerance. So Sylvia is going to do that for abiotic stress. I'm really going to talk only about the drought because that's what I know. So in here, so far we only have focused on Lycopersicum and Pimpinellifolium, but there is so many more tomatoes. So that's what we want to really screen. Like, okay, are there much more cool diversity strategies for all of those different things? So as you can think about, like, drought is a complex trait. If we just measure like, well, do they survive or do they wilt, we're again throwing a lot of complexity into genetics. So how are we going to dissect it? And dissection is going through isolation of individual phenes. So we're going to dissect it into daily use of water, maintenance of growth rate, plant health indices, and photosynthetic efficiency. And that's a combination of the tools that my lab has developed, as well as Phenocyte. So I hope that this is just providing you an overview of what are the tools that are available. And if you want to use some of them, just let us know. And then we can either borrow you some of our cool toys, because that's the best thing of like having cool toys, is sharing them. Or we can help you with setting up your experiment for the Phenocyte. So the first toy that was developed actually by Olga Khmelinsky, who is now a PhD student in plant path, but was an amazing undergraduate student, is this device that's called Osmo. So Osmo is a little Arduino. It's a computer that's costing $20, and we put that in a box with an LED screen, and it's connected to a scale and a little water pump. This is how it looks in real life. So we put all of the mess of the cables in a box, and we connect that to the laptop. Basically, how it works is you hard code all of the reference data for the weight of your plants onto the Arduino. And then you have the plants that are mixed with drought control, and you know that your control plants should be watered to that specific reference weight. Your drought should be watered to, let's say, 100 grams less, right? So you put your plant pot number 1, and it's already hard-coded that pot number 1 is control. It says, oh, okay, your reference weight is below the your measured weight is below the reference weight. Therefore, I need to activate the pump. Pump activated. Okay, fine. Measure the weight again. Okay, now the weight is above the reference weight. Click this button, and then I can do the pot number 2. And so it goes. And if you connect this little device into a Windows-based laptop, because it doesn't work on anything else, unfortunately, but it will also record all of the weight before and after watering, which gives you cheap but dirty evapotranspiration data that you can get. So this evapotranspiration, here I've got an example of cowpea plants that we have exposed to control and drought conditions, and this is the average evapotranspiration rate. We can see that definitely under control and drought stress, we've got a decrease, and we also can see differences between the individual genotypes in their evapotranspiration. So again, this is a very dirty trait, but it's helping us provide to all of the plants exactly the same drought stress treatment, and gets us data that is kind of for free. Then we're going into the maintenance of the growth rate. So for this, we have built something that's called the PhenoCage. So as long as you're not working with Arabidopsis, turns out that plants have 3D architecture, and you kind of have to like image them from all sides. So what we did is we did exactly that. So we're putting a plant by hand on this little display rotating ring that we bought off Amazon, and it's meant for displaying jewelry in the shop. But it's also doing a great job at displaying plants in the growth chamber. So this is the picture how the plant looks like. And again, this is an example of the cowpea, because the technician that is starting the work on the FFAR started this week. So this the plant is turning, and we're taking 7 consecutive images, and we're doing it every 3 days or so. And then afterwards, we're analyzing the images. Because we've got a nice white background, we can quite easily separate the plant bits from the background bits and have that as a projected shoot area. So the number is totally artificial, but it does give you the increase in the size of the plant throughout the time in a non-destructive way. And you can absolutely differentiate. Again, the control is blue here, the drought is red. You can differentiate between your control-grown plants and drought-stress-grown plants. Based on this data, we can calculate the growth rate throughout the experiments for each of the genotypes, which we have done in here. And again, this is for cowpea. And you can see like, okay, so here I've got overall my population kind of decreased by 50%, but I can also do it genotype specific, and I can say, okay, which genotypes are performing relatively well, so close to 1, or some of them are above 1, which are suspicious, and which of my genotypes are extremely tolerant. And this whole setup in general is costing like $200 to build up. So I built it up before we had the big fancy phenotyping facility set up, because we needed to collect some data. But it's a very cost-effective way of getting the data out of your natural diversity panel and identifying which genotypes are of high interest, right? Then we're getting to fancy stuff. So screening for plant health indices. So we're able with Phenosight. So this is using our fancy cool phenotyping facility at the BTI. So we're able to, using a multi-spectral camera, we are able to isolate the NDVI indices as well as anthocyanin and chlorophyll index. So here you've got a tomato plant growing, you've got the color images, you can see the NDVI index, chlorophyll index, and anthocyanin index, and you can see that they are changing at the different time points. And I've got an example of a experiment that we have done with tomatoes exposed to 2 different treatments represented by 2 different colors. And you can see that chlorophyll index, anthocyanin index, and NDVI are showing a very different pattern throughout the time. So again, really, and some of them are somewhat related. So the anthocyanin and chlorophyll index are showing some similarities, but not always, because there is this very early time point that this guy goes up and here nothing is really happening. So you're really able to look at the plant as if through different angles and different strategies, isolate those different strategies more effectively. Then we've got things that are having to do with chlorophyll, fluorescence, and photosynthesis. So I don't know how about you, but my photosynthesis was rusty before I started to work with this machine. So I'm going to explain it to you. So what is happening? We're able to only extract the data that is related to photosystem 2. So photosystem 2 is the one that is like absorbing most of the light. So what's happening? You've got a lot of electrons going da-da-da-da-da. What we need to do first is we need to adapt our plants to dark. So all of those electrons are like, can chill the freak out. So they chill out for 15 minutes. And then we put the plant under a camera. And then we put a tiny flash, and then we can measure the minimum fluorescence. And then we put them under like a super saturating flash, so we can measure the maximum fluorescence. So this is the maximum amount of the light that the chlorophyll can like shine back at us. Fine. Maximum minus minimum is FV, which is the variable fluorescence. FV over FM is quantum yield max. So maximum yield of your quantum yield of photosystem 2, right? Cool. All right. Then what we're doing is we're switching the light back on under the camera. We let it adapt. We see a Kautsky effect, and that's basically related to all of those photons are like either like shown back, or slowly they are being integrated into the photosynthetic machinery, and that's whenever they are, that's why we're seeing this drop in the chlorophyll fluorescence. Then again, we're adding this like super saturating flash, and then we're measuring again, minimum, maximum fluorescence. And again, we can quantify the quantum yield, but it's not maximum quantum yield, because you can see this quantum yield is so much smaller. So once we've got that, we can calculate non-photochemical quenching based on the differences from our light-adapted chlorophyll fluorescence and dark-adapted, and photochemical quenching based on all of the other stuff. So there's a lot of data that we're collecting, and we can do ratios and all of this cool stuff. Anyways, so long story short is, and again, we've got images of color, and then we can superimpose all of those calculations onto individual pixels in the plant. And we can see like, oh, but there is something happening to those leaves that are really suffering at some point. And how is that looking? How is this suffering looking at different traits? And again, we're seeing very, very different trends. So in here, again, we've got experiment of the tomatoes over the time. And you can see that the FV over FM is differing at some point, but then it comes back. So that shows you really the value of looking at dynamic systems and looking as close as possible, because otherwise, if we would measure only here and here, we would be saying like FV over FM, not changing. But because we're looking through time, we can see like, no, there's actually things happening. FQ over FM, which is the light adapted quantum yield, it's not really different whatsoever. And we can see that non-photochemical quenching, there is some variation, and there are some points where this is significant, the differences between our treatment, but the data is a little bit messier here. But again, it allows us to dissect all of those responses into individual categories. And for me, I'm not interested in learning more about NPQ, FV over FM. For me, as a root biologist, I use those to indicate, is my plant okay? Is my plant doing well? So this is my fitness, rather than waiting and growing my plants all the way to the seed and counting the seeds that my plant is producing. Because as I told you before, natural diversity panel, that thing is complicated. But if I do that at the same moment that I'm looking at my roots, maybe this can be a useful data. So before, I have 3 slides left. So just summarizing the phenotype features and capabilities, we can image up to 1 meter tall plants, which is 3 foot for the people that don't like metrics. We have a capacity of 64 large plants. So we have grown citrus trees, we have grown tomatoes, we have grown beans, we have grown setaria. Setaria is a tricky one, but we can. And we have grown Arabidopsis. And for Arabidopsis, we can actually grow 20 plants in one of the tray, which increases our capacity to 1,280 plants. So we have RGB imaging that is masked with chlorophyll fluorescence, which is allowing us to do automatic calculation of the plant size and the growth rate. We also have photosynthetic efficiencies, indices, and high precision watering and fertilizing. So we don't need to come and water those plants because the machine does it for us. How cool is that? So how do you get to use it? I know how to use it. So I left a couple of stickers next to the coffee area. So if you go on the website, there is a project intake form available. So you can download it, you can fill it up to as much as you can. Sometimes you don't really know what you want, but it gives us a good starting point for a discussion. So once we've got this form filled up as much as possible, then we've got a meeting. So it's the person that is doing the experiment, it's me, and it's Julie Bell from the greenhouse facilities and BTI, just to kind of make sure that the greenhouse facilities can provide the conditions that you need for your experiments, that we are not over promising you so that we can track the movement of the microbes in your plant. We cannot do that. So then once we've got that done, we've got a project agreement proposed with the costs. We grow the plants within the dedicated BTI phenotyping facility. And what we provide you with is your raw data, but also some level of pre-analyzed data in R. So basically the same graphs that I showed you in the previous slides, where you've got the changes throughout the time and also those p-values, like when are they starting to be significantly different from each other. We already have pipelines for that, so you don't have to figure it out. And we can keep on collaborating, like whether you want to have something else. Maybe you want to have like 1 leaf analyzed in your experiment rather than the whole canopy. We can also provide you with the snapshot data. So if you've got plants growing preferably in the greenhouse of BTI, because we have had some insect introduction from the Guterman recently, so we would like to avoid that. So if you grow the plants within the facility, you can come in and we can open the machine and just make 1 snapshot data individually. Riley has done that, so if you've got any questions for her user experience, please come and talk to her or me. And then kind of summing up my entire talk of today. So some few take-home messages. So I think studying stress-resilient plants like wild and cultivated tomato can provide new strategies for improved performance under very bad conditions. So you have prioritizing different components of root system architecture can be combined to further improve the tolerance. And also there are new ways, we're working on the new ways of quantifying the roots because we looked at those natural diversity panels. And we have not observed Christmas tree, broomstick, and the telephone pole in Arabidopsis. We only have started to think about it. How are we going to describe those root system architectures by working with tomatoes? I hope that I showed you a couple of cost-efficient phenotyping tools that you can either build yourself or we can help you build them with you to identify drought stress responses, but they can be used for identifying any responses. If you're working on pathogens, probably also quite useful, or nutrient availability. And hopefully with the phenosite, we can provide you with a little bit more depth in your environmental response and quantifying that environmental response. So this is all that I wanted to mention. Of course, this work wouldn't be possible without the amazing lab members that I had, plenty of fantastic collaborators, and also I wanted to give a big shout out to the BTI Plant Growth Facilities team. They have been very, very great in helping me deal with the monster that the phenosite is sometimes, and it's sometimes giving us a lot of headaches, but we're figuring out together, and we're making it work. And of course, the work on the pimpinellifolium initially started in KAUST in the group of this guy, Mark Tester, and I was lucky enough that I could take a lot of it with me. And thank you for your attention, and I'm happy to hear any questions.
I have a question. I have a couple of questions. I can say we don't really know how water is taken. So my question is, what do you mean by that? And the second question is, what role are you looking also at the role of the endodermis, exodermis?
Absolutely, yes, great questions, yes. Yeah, so yes, I will repeat the question. So the question is, what do I mean by the fact that we don't know how water is being taken up? And the second is, what are we including endodermis, exodermis in our water calculation? So both of the questions are pertaining in the cross sections that we're collecting of the lateral roots and main roots, and including them in the Pareto front. So yes, we are including endodermis, exodermis. By the time that I was making these figures, we haven't done the stainings yet for the superization. But yes, we are. We know that the exodermis is forming first, endodermis is forming later. So we are including them in the tomato. Nobody has ever looked at the exodermis and endodermis formation in the lateral roots, which is mind-blowing to me that nobody has done that. But knowing how difficult it is to get those lateral roots cross sections, I start understanding why nobody has done that, because those roots are very finicky. So we're embedding it in the plastic resin, which is as you probably know, a bit of a pain.
Can you stain the whole without cross section?
Can we stain the whole root without a cross section? It might be. We could potentially, but because we already have the cross sections, might as well just stain those cross sections. So I answered your second question first. Now let's go back. What do I mean by we don't know how water is being taken up? Well, we know roots. That's short answer. But if you think about, okay, so, and I have been having a lot of conversations with Guillaume and people in Louvain that are developing a lot of water transport models. So you would think that they know. The question is majority of the water being taken up by the root tips or throughout the main roots? So majority of the models are actually developed using monocots. Monocots, they have what the vasculature is changing and the conductivity is changing. So you've got axial conductivity and radial conductivity and axial conductivity, right? So radial conductivity is decreasing with the age of the root, and axial conductivity is increasing, right? So the older roots are kind of becoming more pipes rather than absorbent, right? And that's for monocots, and the monocots don't show any change in the root diameter. Dicots, on the other hand, we do have secondary growth in the dicots. So they have, and Marco is a PhD student of Guillaume. Marco hates me for that because I forced them to develop those models for tomato. And they're like, well, in dicots, we've got secondary growth. So your diameter is not changing. So even though you've got axial conductivity, radial conductivity decreasing because you've got those endodermal things, you've got more woody stuff happening, and so forth, your diameter is increasing. And the increase of that diameter is sufficient for kind of counteracting that effect that we're seeing in the monocots. So the thick roots and dicots are absorbing just as much as the thin roots, according to their models. So we're trying to kind of see what's happening if we what can we learn from the model into the real life, and can we just grow plants with thick roots and just chop off those main root meristems because we apparently don't need them, but I think we still do. So that's what I mean by we don't really know how the water is being taken up, because we don't know what features are the most. I mean, we do know that features like cortex are not, number of cortex layers, it's not really playing much of a difference. The number and the size of the xylem vessels is. Endodermis, exodermis is absolutely playing the majority of the big pictures. But we don't know how it's working with the root diameter is increasing, because all of the sudden things are being like, it's not, yeah. And it's in the way, it's good that they developed all of those models on monocots, because it's a simple system. And now we're upgrading into the dicots.
Yeah, sure. I had a question about the two tomatoes and the cross.
Yes.
I was wondering, was there any differences in associations with mycorrhizals? So mycorrhizal associations with in the cross, does that change it?
Okay, so Claire is asking whether there is a difference between the mycorrhizal association. We have grown those plants in the sterile conditions on the agar plate, so we don't know at all with the mycorrhizal associations. They are performing both equally well when we grow them in the soil. We don't incubate any mycorrhizal communities at all, but I assume that they just emerge. I don't know anything about mycorrhiza, so I see potentially there's definitely potential there, but we haven't studied it yet. So if you want to have some seeds, we can totally give you. Sam.
I was wondering, so you have the differences in the root architecture.
Can you speak up? I can't hear you.
In the hybrid line, do you see a difference in the Ross accumulation?
We have, so Sam is asking whether we see differences in the Ross accumulation in the F1 hybrid. We have not looked at that because we had so few seeds of the F1 and we haven't sent it to Ron because we were like, well, how about we establish this protocol? Because they haven't done the roots before, because they were like, oh, roots, there's a lot of autofluorescence. We don't do roots, we only do shoots. I'm like, roots are cool. So, and we see huge differences in the stress response between the roots and the shoots. So hopefully now we convince them. But that's a great idea to do that on the F1. I don't know. And we haven't done RNA-seq on the F1 as well, which would be another way of kind of helping us figuring out not the cis and the trans of it all, but also like which of the responses are dominant from which genotype. Yeah, great idea. Thank you. Yes, John.
So how would you determine the change in architecture is actually adaptive, like for salt tolerance?
Yes.
Like how do you know that this change is
Okay.
not a consequence of stress response?
Yes.
Damage.
Yeah. Okay, so Jan is asking how are we going to identify whether change in the root system architecture is an adaptive response? We don't know yet. So we're assuming that natural variation, and we assume that the maintenance of the growth is something that is adaptive over the long term. Because if you've got roots that are growing, and I think Jose Dineni published on that, if you've got roots that are kind of keep on growing throughout the stress exposure and they don't stop and think, they typically, the meristem activity is decreasing afterwards. So we are focusing on the maintain meristem activity, and hopefully that is allowing us to then say like, okay, well, if you've got, after a week of stress, if you still keep on growing, it means that, okay, you can survive that. And we're also going away, we're working with the stress concentrations that are as physiologically relevant as possible. So we're working on 75, max 125 millimolar salt stress. So we're trying to stay away from osmotic shock because I think that's also not how salt stress is typically occurring in the wild. But yeah, we don't know. And that's also one of the reasons that we want to add that a lot of the stuff that I have presented is a very descriptive studies. So we are going to identify the genes, and once we're at the genes, then we can say, is it adaptive, is it not adaptive? But yeah, a very good question, and it's definitely something that we have to keep in mind. Like the fact that things are changing or like stopping growing, it's not necessarily a good thing, but maintenance of growth, we are assuming that that's what the plant respond.
Just some time, so let's thank.
Thank you so much.
This has been a production of Cornell University, on the web at cornell.edu.